If you are asked to give some supporting points, maximum 2 points you should write in support of your identification, and one special site should be given. You should draw the diagram and you should mark it. Now, at the time of drawing, suppose muscle slide was given. You draw actin and myosin, you draw, and you mark it actin and myosin. It is ultra structure. You are given light microscope and you are writing findings of the electron microscope. This should not be done. Only light microscopic feature should be done, should be written, right? Now, in case of suppose one muscle fiber was given, muscle slide, skeletal muscle. 4 or 5 points you should write down. Number 1, the main points is, in case of skeletal muscle, nucleus. Skeletal muscle contains multiple nuclei. Multiple flattened nuclei peripherally. Number 1 point, major point. Number 2, there is cross striations. Cross striations, very prominent cross striations. It is the characteristic of muscle, skeletal muscle. Number 3, there is some negative points also. As for example, the muscle fibers do not branch. There is no branching, right? Third number point. These muscle fibers, you know, each muscle fiber is a single cell. Though I am calling it fiber, but it is a cell, single cell. Now it is covered by single cell. Outer member of the cell, what is the name of the outer member? Plasma membrane, plasma lemma. And outside plasma lemma there is endomysium. There is a connective tissue layer that is called endomysium. So each fiber is covered by endomysium, right? Now several fibers, when will be grouped together, they will form fasciculus. That fasciculus is surrounded by perimysium. When fasciculus should be surrounded by the connective tissue, this is known as perimysium. Several fasciculus will form the bundle, muscle fiber that is surrounded by epimysium. Endo, peri and epi, right? So these are the, and if you can remember the length of the muscle fiber, you may write down, that is the few millimeter to few centimeter in case of muscle fiber. In case of cardiac muscle, it is about 80 to 120 micrometer, and in case of smooth muscle, it is about 15 to 500 micrometer. So it is not very important. Important is the number of nuclei, right? Now come to the cardiac muscle. Cardiac muscle, the nucleus of the cardiac muscle is single nuclei. Cell shape of the cell is cylindrical, short cylindrical. Nucleus is central in position, oval in shape. Here also you will get striations, but it is not so prominent as skeletal muscle, right? Here the most characteristic feature of cardiac muscle is it branches. There is branching. You should see that there is branching of the muscle. At the end you should see the branching. And another important characteristic, that is intercalated disc. The adjoining muscle When two muscles are joining, there you will get intercalated disc. Right? Zigzag intercalated disc. One question is generally asked, do you know what is syncytium? Syncytium? Multinucleated protoplasmic mass. Multinucleated protoplasmic mass. So one question is asked in case of heart muscle, that is it a syncytium? If one muscle fiber is stimulated, the whole muscle will contract. That means there is connection. Is it syncytium? Actually, anatomically, there is definite cell membrane. So anatomically, it is not syncytium, but physiologically, it acts as a syncytium, because I talked about intercalated disc. Intercalated disc, actually, here there is desmosome, tight junction and gap junction. Through this gap junction, impulse will pass. So it acts as a syncytium. So it is called physiological syncytium, not anatomical. Right? And these are the points in case of cardiac muscle. Now in case of smooth muscles, smooth muscle is spindle shaped, long spindle shaped, and it also contains single nucleus. Single nucleus at the central position, the maximum diameter, where there is maximum diameter, there will get single nucleus. And here, as it is called smooth, so there is no cross striations, right? And if we consider, though it is not a histological picture, but you may be asked in the exam, what is the nerve supply of the skeletal muscle, nerve supply of the heart muscle, and nerve supply of the smooth muscles? Smooth muscle is supplied by autonomic or somatic? Which? Autonomic. Smooth muscle is supplied by autonomic nerves. Likewise, cardiac muscle is supplied by the autonomic nerve, but smooth muscle is cerebrospinal nerve or somatic nerve. Right? So these are the, now come to the smooth muscles. The smooth muscles point is spindle shaped, centrally placed nuclei. These two findings similar to the heart muscle, cardiac muscle, but the differentiating point is in cardiac muscle there is intercalated disc and slight or cross striations. There is no cross striations, right? Cartilage. Cartilage are of three types: fibro, hyaline, and elastic. Hyaline cartilage. Hyaline cartilage, actually hyalos means glass. As it is very transparent, intercellular substance is very transparent. That's why it is known as hyaline cartilage. Now in H10, the hyaline cartilage, the cells, in case of cartilage, the cells are separated by a intercellular substance. Intercellular substance looks transparent, and in H10 they take blue staining. And within this intercellular substance, what you will get? You will get fiber. What type of fiber? Collagen fiber. That collagen fiber lies parallelly. In case of hyaline cartilage, you get within intercellular substance collagen fiber, and intercellular substance, and it is the matrix. Intercellular substance means matrix plus fiber. This matrix contains chondromucoprotein. Matrix का main constituent है chondromucoprotein. Now the sites. What are the sites where you can get this type of cartilage, hyaline cartilage? Where you will get? Synovial joint. Synovial joint is a broad thing, but specifically you have to mention articular cartilage. Articular cartilage. And no, pubic symphysis not. Costal cartilage. Growing bone, in growing bone you will get epiphyseal cartilage. Trachea, bronchi, thyroid, they are called or hyaline cartilage. And if you go above means above the thyroid cartilage, you will get cricoid cartilage. Above that you will get epiglottis. Epiglottis again it is not hyaline cartilage. Epiglottis, corniculate cuneiform and arytenoid cartilage. There is small cartilage, arytenoid cartilage, the apex of the arytenoid cartilage, they are all elastic cartilage. In case of pinna and external auditory meatus, it is less elastic cartilage. Pharyngotympanic tube, you will get outer covering, that is pericardium. But in case of fibrocartilage, there is no such pericardium. It is one of the differentiating point. In case of hyaline cartilage, you will get group of cells within the intercellular substance and a group of 2, 3 or 4. In the central position, you will get more cell than peripheral position, and this group of cells is known as cell nests. So you should write this slide contains cell nests. Here you see group of cells. Here group of cells. It is group of cells. Here there is 1, 2, 3, 4. These are called cell nests, right? These are called cell nests. And if you go outside, there this thin layer is called perichondrium. This thin layer is perichondrium. This perichondrium is absent in case of fibrocartilage, but it is present in case of hyaline and elastic. And at central position, you see the population of cell is more and bigger than the periphery. Here you see the cell is bigger and population is more than the periphery, right? And intercellular substance, these are transparent. Here, though it is not actually bluish in color, but here it is not looking like that. But in good side, you will get bluish color. The intercellular substance will take hematoxylin stain, that is bluish in color, right? Here you see the cartilage cells. Actually, hyaline cartilage, the intercellular substance contain elastic cartilage. In case of elastic cartilage, intercellular substance or matrix contains elastic fiber. They are not parallelly arranged, but they form a network, haphazard direction. So they form a network, and on HE stain, they do not. Actually, this slide doesn't take HE stain properly. To stain elastic cartilage, special stain is used, but we are here seeing the HE stain, right? And now here also you see the cell nest. Here cell nest contains maximum a group of 1 or 2. There you found in case of hyaline cartilage group of 4. But here is there any group of 4? Any single? You see? There was single, sub-single cell, a single. There is no a maximum group of 2 or single, a group of 2, right? So, in case of hyaline cartilage, you will get cell nest, but the population of cell either 1 or 2. Right? Here you see at the periphery there is capsule. This is capsule. So it is elastic cartilage. Now you will be asked where elastic cartilage is found. I already told you, pinna, outer part of the or external ear, inner part, medial part of the pharyngotympanic tube, and epiglottis, uniform corniculate, eriepiglottic, arytenoid, apex of the arytenoid. These are the sites where you will get elastic cartilage. Can you see multiple nuclei? Is there any multiple nuclei? From distance you can see multiple nuclei. Nuclei. Here you see there is nuclei. These are some nuclei. These are nuclei. Right? Actually, it should be arranged very fairly, but here it is haphazardly oriented. And there is no branching. If there is no branching and if there is multiple nuclei means it is skeletal muscle. Only one muscle contains multiple nuclei, that is skeletal. If you see that hematoxin is 10, these are nuclei. These are nuclei. If there is multiple nuclei along the periphery, you will diagnose it with a slide of skeletal muscle. Right? Here you see skeletal, the nuclei are not peripherally situated, haphazardly situated. Actually here we cannot find the branching. Right? There is no branching. I think it is cut transversely. So it is not a good slide for you. If we get good slide, you will be again shown. Right? Just theoretically, you know, intercalated disc, branching, single nucleus, centrally placed. These are the points. Right? So these are the muscles on the other side. You should draw it in your histology book and it should be properly signed. Otherwise, you will not be allowed to sit for the exam. Right? What type of superficial layer? What type of cells? Umbrella shaped cells. But you have drawn picture. Superficially, you have drawn that squamous epithelium instead of umbrella shaped cells. So how will you get marks? Then you should write where it is found. Okay, one question. Explain why. Clavicle is a modified long bone. First of all, you have to write why it is called long bone. Long bone means long bone's function it transmits body weight. The clavicle also transmits body weight. It carries the weight, transmits the weight of the superficial upper limb to the axial skeleton. So it is acting as a long bone. But long bone is not generally transversely placed. It is generally longitudinally placed, vertically placed. Then you should write all these things. That's why it is called modified. Femoral hernia is more common in female. Only one line. If you write only one line, because of the wide pelvis, the femoral canal also wide. Because of this wide canal, more chance of hernia. In case of even only one or two lines. Carcinoma of inferior quadrant of the mammary gland. From inferior part of the mammary gland, some lymphatics pierces the rectus sheath and makes connection with the subperitoneal plexus. From that subperitoneal plexus, through the peritoneum, that is translumic root, it invades the ovary. Krukenberg tumor. This is called Krukenberg tumor. Right. Though there is some controversy that Krukenberg tumor, if it is translumic spread, then the tumor should be found at the peripheral part of the ovary. But it has been found, these tumor cells, malignant cells, are found at the center. So someone say it does not spread translumic route, but it spreads through the hematogenous spread. That's why central part has been involved. Why conceptus is not rejected by its mother? Actually, conceptus means the developing embryo. You know, when the embryo is developing, there is trophoblast, cytotrophoblast. Around the cytotrophoblast, there is a fibrinoid layer. That fibrinoid layer actually covers the whole thing. So it actually preventing the antigen of the fetus to come in contact with the mother's immune system. That's why it is not rejected. This is the main point. Right?